Welcome to MSB Talks, a podcast about science for everyone. Our guest today is Dr. Julie Biteen, who is currently the Janine Maddock Collegiate Professor of Chemistry and Biophysics at the University of Michigan. Her lab develops single molecule fluorescence imaging techniques to study biology and material science at the nanometer scale, with applications spanning microbial cell biology and metal nanoparticle plasmonics. This episode, we talk about Dr. Biteen's journey through her scientific background, research interests, and thoughts of the future of single molecule imaging. This is MSB Talks. We're glad to have you. And now for our conversation with Dr. Biteen.
Hello, how are you doing?
I'm great. I just gave a lunchtime talk to our REU students, so the summer research program in biophysics. So
Nice.
Nice.
It's fun.
We also just did an interview, so we're both
Yeah,
warmed
we
up.
just did one right before this. So yeah, we're warmed up. Exactly.
it's right in the mode.
Yeah.
Yeah.
All right. Well, nonetheless, thank you for giving us the time to interview you. We appreciate the opportunity to talk with a professor. Um,
Yeah.
but yeah, I think we'll just maybe introduce our project first, and then we'll
Perfect.
hop into questions. All
Yeah.
right. So. Our project kind of revolves around this idea of what we call bacterial drug delivery. So effectively, we're transforming a non-pathogenic bacteria into a delivery vehicle for a cytotoxin. So one cool thing about the bacterium that we've chosen, it's called AMB1. Um, it's first chemotactic and aerotactic, so it naturally navigates to low oxygen environments, such as like a tumor microenvironment. And secondly, it's magnetotactic in the sense that if you hold it up to an external magnetic field, it can move along those field lines that you set. So those are like two kind of core screen strategies that we can use to localize our bacteria to our area of interest. Um, but beyond that, what we're doing is for each bacterium, we're constitutively expressing um, a surface protein that's meant to bind to a receptor that's commonly overexpressed on, a given number of conditions. For us, we're working with ovarian cancer and folate receptor alpha. And the goal is essentially using those two taxis strategies for tumor tropism, um, and then using like this surface protein to have the bacteria physically dock to the tumor, and then using a heat shock promoter and just reaching a critical temperature to induce the production of, a cytotoxic payload. One way you've kind of framed this is like an antibody drug conjugate, but without the linker. Effectively, you're just keeping the gene for the cytotoxin silent until you've confirmed um, localization. And a big part of why we reached out to you is that one of our plasmids that we're using in our system, it encodes a fluorophore called mini SOG, which is a fluorophore that can fluoresce in an anaerobic condition. Um, we honestly aren't like too familiar in regard to like fluorophore design and function, but we did like some literature research just to kind of determine whether this would be a viable fluorophore. Um, but yeah, no, we would love to pick your mind about that and other thoughts on our project. But yeah, I guess before we hop into like more technical stuff, we would love to learn a little bit about you and what exactly you do.
Yeah, so well, my name's Julie Biteen. I'm a professor in chemistry and in biophysics. And so what my lab does is develops super resolution microscopy and quantitative fluorescence microscopy to look at really organization and cooperativity, molecular scale things inside living microbes. So, as far as bacteria go, what we're really proud of doing is looking at single molecules of, let's say, a protein of interest, watching how they move in the cell. And we could detect things like binding or enrichment at a condensate based on the fact that, the molecule is freely diffusing and then it's less or more confined. And so that's sort of the technology that we're working on. We have a lot of different applications, but all around that type of technology.
Cool. I guess could you just like maybe talk about your like background in research, kind of how you got to where you are. Yeah.
Yeah, for sure. So I actually started, well, my PhD was in solid state physics. So for me, the thing that's really been a joining factor has been optics. And so what I was doing was nanomaterials and optics. So in my PhD, I was doing a lot of quantum dots, which is when you take a semiconductor and you make it really small such that it can interact more efficiently with light, and plasmonics, where you're coupling those quantum dots to metal particles to change the electromagnetic fields. And so I really like that, but was looking for more applications, sort of health related applications. I was really looking for applications that you can I don't know, call grandma and tell her what you're doing, or, connect to what's in the news. And so I was really excited to transition towards more biophysical chemistry. And so I did a postdoc at Stanford University with W.E. Murner, who later won the Nobel Prize for this super resolution technology. And learned a lot about microscopy and imaging, but also was able to learn how bacterial cell biology works. So I come really from the more solid state physics, physical chemistry side, and then have become a bacterial cell biologist sort of by osmosis over the years.
Yeah, that's awesome to hear. It's not common that like faculty will make that large of a jump, especially after like completing a PhD. So it's really cool that you're able to like, kind of shift your area of focus. Um,
Michigan's a great place for that too, though, because there's such an emphasis on multidisciplinary science. So what are y'all's majors?
Molecular biology and philosophy.
Uh,
Yeah.
Biomedical engineering.
And so, you could come from all these different points of view and then still impact, your shared project. And it's actually really helpful to have those different backgrounds when you work on a team. And so in my research lab, that's sort of been the approach. It's like, well, I didn't actually ever take a very rigorous biochemistry class, for example, so there's things that I don't know as well, but then I have some students who can, really school me on that. There's always a student who's really good at statistics, or a student who's really good at coding, and, just having those different backgrounds is a strong asset at Michigan.
Yeah, for sure.
Yeah. Yeah, no, I definitely agree that especially when you're working like in a team or in a lab, like it's important to have people from a variety of disciplines and backgrounds. Like, for instance, our group is like I think close to 30 undergrads,
Mhm.
but we're split across like biomedical engineering, chemical engineering, computer science, physics, biophysics, math, that kind of thing.
Mhm.
Yeah.
Um, but yeah, I guess more broadly, what topics excite you in today, I guess in imaging and microbiology?
Yeah. For me, and actually I was telling you, I just gave a talk to our RU students. And so something I'm really excited about is a common theme that's been emerging across bacterial cell biology. And, for a long time, I've been very interested in how bacteria cells are organized. Obviously, bacteria cells don't have membrane-bound nucleus or, mitochondria or things like that. But there's definitely localization, compartmentalization, and that drives my research program. So over the last 10 years, the language, but also the physics context that we've been thinking about this in is really the idea of phase separation and condensation. And that's something that's really exciting to me. So, you all are thinking about taking the bacteria with a payload and getting it to a target. And what I'm thinking about is what if you zoom in, 10x, and now what you're looking at instead is taking a protein in a cell and getting it to where it can affect its enzymatic function. And so one of the ways that it's emerging that proteins can be localized, concentrated, interact with their substrates is through this idea of forming membraneless organelles, liquid-like condensates. And so we've been looking, on one side, my lab thinks a lot about technology. And so we've been looking at how do I know something's a liquid? and I teach freshman chem. So, freshman chem, you're like, okay, well, in a solid, the molecules are ordered and they can jiggle a bit, but they don't move. In a liquid, they move more, but they can be contained. And so when you do single molecule tracking and you can think about that, we're actually able to ask, are the molecules inside the cell moving? Are they confined but moving, or are they stuck? And you can actually differentiate between those two things. And then the reason I think that's important is because any kind of diffusion limited reaction is going to be affected by the material state. And so we've seen, for example, transcripts that can find the spot where the RNA degradation enzyme is located, but can't get degraded because everything's so solid-like in that state. And so the stress response, the adaptability, those are things I'm really excited about these days.
Yeah, that's awesome. I guess kind of going back to kind of applications of like fluorescence that kind of applies to our project. I mean, we like our goal is to get the bacteria into tumor cells, excuse me. And so they, and we use fluorescence to actually see if that's if that has been achieved. Um, and I guess like from your work with like, imaging these bacteria, like what would you say, like what makes a fluorescence signal actually like, kind of trustworthy as evidence that it has made it to where it needs to be? And how can it sometimes like how could we, as student researchers, how could we mess that up? Or how could the fluorescence signals kind of mislead us?
Yeah. Tell me a little more about what you're doing. Are the tumor cells also labeled? How do you know? You can see where your bacteria are. How do you know the context?
Yeah, so our goal right now is like once we have our bacterial system developed, we'll co-culture it with a high-grade serous ovarian cancer cell line. And after that, we'll use confocal microscopy to kind of figure out where these where our bacteria are going. So our bacteria will ideally have like some kind of fluorescence to them because of that fluorophore.
Mhm.
Um, and we've been thinking about how to kind of approach this with like the cancer cells because obviously labeling them is an effort in and of itself. But one other thing that we've been thinking of is that if you just take like a 2D cell culture and you just grow cancer cells in them, they tend to form like foci on the dish. So if we can somehow find out and pinpoint where those foci are, then if we confirm that our bacteria are fluorescing around those foci, that's kind of like an indirect validation that they are colocalizing.
Yeah, so I mean, you're speaking to one of the major challenges, which is almost nothing that you're ever looking at is naturally fluorescent, right? So everything is going to be label based or some sort of contrast based. And so I guess you're taking advantage of some some feature of your cells that the tumor cells are darker or something like that. And so now you could say, okay, the bacteria cells colocalized with it. Um, and that's, so that's the first thing. But then you sort of, without saying it, you basically said it's a proxy for
Yeah.
colocalization because if two things are close, what that literally means is they're close. And
Mhm.
so how do you know that they're mechanically touching, chemically touching, things like that, right?
Mhm.
So a couple things that I think about, one thing about this sort of co-localization aspect is maybe a question of reversibility. And so, you can imagine if the bacteria cell swims over your cell and doesn't, it happens to be there, but it doesn't interact, it just keeps moving. But if it stops and, there's a lag, and so you could think about the binding time, the interaction time of that event. Those types of signals I think are really helpful. And from an image processing point of view, that's not that hard because you're cancer cells are actually static. So really what you're saying is, is a bacteria moving? Can you track it over time? Can you see it stop? And does that stopping, that temporal information, does that tell you also that's the time when you see the colocalization in the microscope? The other, the other problem that we always have when we're thinking about labeling is the label specific? And so in this system, it probably won't be a huge concern. But you can imagine, if, for example, the bacteria cells are very weakly fluorescent, such that, I don't know, just some dirt, some dust, something that gets into your microscope is scattering about the same amount, then, then you start having these false positives and have to start validating it. And so you're using a plasmid to generate the label, and it's going to be intrinsic in the cell. But the other thing is, if you use an extrinsic label, sometimes you have to think about, okay, I labeled my cell, but is there excess dye, and where is that going? So I think those questions of the false positive at every scale become an issue. But it seems like in this design, it shouldn't be such a concern.
Yeah, I guess sorry, what sort of controls would you want to see, if you're looking at some sort of data, before you actually believed co-localization, what controls would you need to see personally in someone else's research before you believed it?
So you talked about how your cells have some sort of sticky groups that you think is why they're going to stick, for lack of a scientific reason.
Yeah, so I think
Yeah.
we've read like in literature that at least it's something that we're using. Cancer cells like lack contact inhibition, so in that sense, like they'll just grow on top of each other. And we're just relying on that to be the case.
How about the bacteria adhering to the cancer cells?
Oh yeah. Those have like a surface ligand, which is meant to like bind to folate receptor alpha, which is overexpressed in, I believe, 70 to 90% of ovarian cancers. And in the cell line that we have it, it overexpress folate receptor alpha, so it is antigen positive.
So the one obvious control, if you think about it that way, is you have some cells that don't express that group. And the answer might be, some still stick because stuff sticks. But, so what I would want to see is 1% of cells 1% of the cancer cells have a bacteria cell stuck on top, but usually it's 1 or 2, versus in my real experiment, 55% of them have 10 or more bacteria stuck on top. So just seeing that difference and seeing what the noise level is of the experiment.
Yeah, that makes sense.
Yeah, and kind of building on that, do you think it would be like a plausible control to maybe like first incubate our bacteria with the cell line and then maybe like do something to try to wash it in a sense? Like maybe add like yeah, this might be a little tough because I'm trying to think of it in terms of like how you would approach like a blot, for instance, like when you're incubating like a membrane and an antibody, um, you'll just wash it directly after to get rid of any non-specific binding. Do you think there's anything similar that we could kind of think about here?
I guess, I mean, I have a trite answer, right? Which is try it. Yeah, and I mean, this is similar to experiments that are going on in my lab, in that, we add a dye sometime. So, well, okay. So in the experiment I'm thinking of, we have a bacteria toxin that's dye labeled, but sometimes it just sticks because things stick. So then you start asking, how do I wash off the things that are not covalently bound? And so, just try it, right? So then you try it, and then actually it washes off everything that's covalently bound too, because you're doing too good a job. So, the question is, can you find a happy spot? At some point, maybe something as boring as just, rinsing your surface with some fresh media will do it.
Yeah.
And so, why wouldn't you try that? But the specific problem we had when we did something like this, and it wasn't with cancer cells, this was just bacteria cells and proteins, but The specific problem was my student did too good a job at washing it and everything washed away. Now you have no signal. So, you know,
Yeah.
think about that balance of forces.
Mhm. That makes sense.
Yeah, so a lot of a lot of the work that we saw from your lab is done in like controlled conditions, right? Like you
Mhm.
have bacteria in a dish and then you incubate them with a toxin or a dye and then you just essentially see what happens visually from there. But something that I mean, and obviously we're for the foreseeable future, that's what we're going to be doing too. Um, but one thing that we'd like to do eventually is to take our bacteria and perhaps put it into like a more representative system, whether that's like an organoid or probably not an animal model, but maybe if this project
Yeah.
passes, um, or something like that. Um, and obviously that kind of environment poses like not only like a spatial issue in terms of like, you're adding a dimension, but you're also, you know, adding more elements into the mix than you would have in like just a controlled setting. Um, what would you see as like the hardest part of imaging bacteria if they're in like a more, I guess, quote unquote, messy system?
Yeah, I think you mentioned the two biggest things, right? One is so it's very convenient to look at single bacteria cells because they're about a micron thick, and the depth of focus of my microscope is about a micron. So I can see the whole bacteria, you know, and then I can very carefully control my sample such that there's nothing fluorescent, out of focus, no background. And that's how we're able to
Mhm.
see single molecules. The second thing that you mentioned, and so right, so I mean, if it's thicker, it's a problem, and we're thinking about this in terms of walking towards imaging biofilms, for example, which start becoming a whole complex environment. You know, the material can be very scattering, so it's not just that it's thick, but it's also it could be absorbing or scattering, and or even fluorescent. So that's one set of issues. And there's different approaches to optical sectioning. So one thing that we're doing is using a light sheet microscope so that instead of just illuminating the whole sample and getting all that background out of focus, we're illuminating from the side so that only the plane that we're imaging is fluorescing, and we have a lot less background. Um, so there are approaches, but you'd have to think about that. But then the second thing that you mentioned is the complexity, right? And so, you know, you asked me before about controls. The other thing I would definitely want to see with any of these types of experiments is, can you do it 3 times on 3 different days,
Yeah.
preferably 3 of your teammates doing it, different people, right, following the
Yeah.
same protocol. How reproducible is it? And it's totally normal that things change a bit. But if you want to make
Mhm.
drastic conclusions, you want to make sure that the variability from day to day is smaller than the change that you're measuring.
Yeah.
And so I think the more complex the experiment gets, the more factors will broaden your distributions. So I guess
Mhm.
my recommendation is baby steps, right? So you said, no, not a whole animal. I'm like, yeah, you know, the goal could be a whole animal, but where do we go first?
Yeah.
And so a few things that come to mind. I think organoids are interesting, but they're already messy. So, you know, there's these biomimetic type of I don't know, picture like something that's 3D printed. And I see this, for example, when we think about soil microbes. You can imagine it's really hard to do microscopy of bacteria on soil because soil is pretty heavily absorbing. And so you can 3D print materials that have the roughness and the fill
Hmm.
factor, but are clear plastic. So, you know, thinking about, can you manufacture something that's really well defined, and maybe start with that. Um,
Mhm.
and then the organism I think is really interesting is zebrafish. And I think if I was going to
Hmm.
do like bacteria in a multicellular organism, the zebrafish experiments are really interesting. And you could Google bacteria and zebrafish, and you get these really gorgeous images. And again, right, probably there's no tumor cells, but maybe there's some kind of cell with a specific receptor. So you could do the analog and at least show the proof of
Mhm.
principle. And those are transparent, and that's why it's important. So they're
I
multicellular
see.
organisms, but they're
Okay, that makes
transparent.
sense. I
So,
was about to
just
ask why
thinking about
zebrafish
that.
and not
Mhm.
mice.
Yeah.
That
Yeah,
makes sense though.
they're also there's 20 reasons why they're easier to work with than mice.
Okay.
Even from a kind of humanitarian perspective of like, does the research you're doing warrant mammal experiments, things like
Mhm.
that. And then,
Yeah.
but also just like how fast they grow, how
Mm.
expensive they are, things like
Hmm.
that.
Okay. Yeah. I mean, just due to the project, and this is for a competition for the iGEM competition. Yeah. And so we're not allowed to use animal models at all. So that's why, yeah, that's why we said no animal
Definitely not
models
animals.
at
Got
least
it.
for now. At least as long as this project is still for iGEM, no animal models.
Mhm.
Maybe one day it'll get a life somewhere else, but at least for
When you
the
start your
kind of
company with the product.
yeah, exactly. For the short and medium term, definitely no animals allowed, unfortunately. Um, so yeah, we're kind of exclusively looking at just the cell lines that we've been talking about, and then maybe organoids is something that we've heard a lot from from different professors that we've talked to,
Mhm.
have all kind of said organoids are a good next step if you can't
Mhm.
do animals. So that's probably the next thing that we would potentially do. Time and budget allowing, I think. But yeah, so
Excellent. Have you guys been on the team for several years?
I have not. I just finished my freshman year, so I've been here I started in like what, January was it? So I'm pretty new.
Yeah, and this is my second year. And this is coincidentally like I think the first project we've spent more than a year on. Because
Okay.
usually like at the start of every year we just change up the project. But um last year we made some decent progress with it and we really didn't want to like say goodbye to it yet. So, you know,
Yeah.
it was a unprecedented decision.
That's
Yeah.
fun.
Who knows? Maybe we'll go for 3 years. You never know. Um, but yeah, so I guess like kind of a similar question to the last one. Just like when we're thinking about like our bacteria, we said that they're magnetotactic. So they like that they respond to magnetic fields, and we're trying to like use magnets to move them around, to orient them, whatever. Like if we wanted to use imaging to somehow watch a process like that, or like we're also building an AMF device so that we can use a heat shock promoter to get them to produce a protein within the tumor environment. So if we wanted to like use imaging to watch that turn on, or something like that inside of living cells, how would you think about choosing the right imaging approach to do that?
Yeah, I think that the watching the bacteria move in response to magnetic fields is going to be pretty straightforward extension of what you've already thought about, right? Because if you can see the
Hmm.
bacteria, you can see the bacteria. So then
Yeah.
it's just how do you apply your magnetic field in the microscope? Uh,
Hmm.
and I'm sure it's not trivial, but I'm sure somebody's done it. Question mark. Um,
You'd think.
okay.
I mean,
So
yeah. Who knows?
I
But
think maybe if we use like a weak enough magnet, nothing bad will happen. Who knows?
hopefully, yeah. I'm not sure we've fully gone down that avenue yet
Yeah.
of
yeah,
exploring what's been done. But
I haven't,
um,
I really haven't thought about what types of strengths of magnet are needed there compared to what I would be nervous to put on the table.
yeah.
So, I guess that's the question. And probably y'all have called microscope vendors before and asked for advice, that this is a good place to do that.
Hmm.
So, not just read the manual on the website, but call up
Yeah.
Olympus or Nikon or whoever's still do it and just say,
Mhm.
yeah, if it was up to you though, like, I mean, is this really gonna ruin it? Um, so, more in my wheelhouse is how do I know that this sort of response, the heat shock response has occurred? And
Mhm.
so what we think about a lot is taking some protein you're interested in and making a fusion to a fluorescent protein. And so now the the result would be that after whatever induction event happens, so the heat shock, whatever it is, the protein starts expressing. Every time the protein is expressed, you'll also have your fluorescent protein expressed. And so conceptually, I think that is pretty clean, but the
Mhm.
main question is time scale. So proteins usually obviously take some time um, to be expressed, but also to fold. And so, and fluorescent proteins, will take maybe an hour or so, even like a fast folding fluorescent protein, it's still fast relative to hours, not second scale. And so
Yeah.
you'd lose some temporal information, but you would get some information.
Mhm. That makes sense.
Yeah, I mean, that's a lot of what we're doing like computationally as well. Like we're essentially using our computational modeling subteam is essentially trying to fill in the gaps that we can't address in the lab.
Mm-hmm.
I guess it's a big advantage to having like that kind of resource at your disposal. Just like we in studying like the distribution of our treatment, for instance, and like the dynamics of it in relation to like the receptor and the ligand and the broader diffusion of it over a certain time scale. Um, yeah, no, that is definitely something that we are trying to look into. Um, but yeah, no, nonetheless, we really appreciate all of your technical insight and giving us the time to sit down with you. But I guess before we wrap up, we do like to ask all of our guests for a bit of advice, just for anyone who's interested in science or medicine or following a career path adjacent to yours or like yours. So if you could offer us anything, that'd be fantastic.
Yeah.
So the biggest advice I always have is we could all be happy doing a lot of different things. And so it's just really important, and especially in your endeavor right now, but really important to surround yourself with a good team, with supportive mentors, with supportive colleagues. And so, for me, it's really, yeah, I do a lot of interdisciplinary science, and I didn't set out to do biological work, but I found it because I've found the right community. And so, just making sure you're finding community where you can thrive,
Mhm.
not compromising your standards or anything, but don't be too stuck to one idea, and rather just think about where can you be the most successful in the context.
Awesome. Well, thank you so much. Thank you for giving us your
Absolutely.
time. I know you're busy, and summer's a busy time for labs. So
Well,
yeah. So thank you
good
for.
luck with the competition. When is it?
It's
Uh,
in, I believe, end of October, beginning of
November,
November.
November.
November. Okay.
So we'll work on it until then. But yeah,
Yeah.
Hope it goes great. Enjoy the rest of your
Thank
summer.
you.
awesome. You
Yep.
too.
You
Thank you
too.
so much.
All
All
right.
right.
Bye bye.
Thank you.
Bye.
Bye.
That concludes this episode of MSB Talks, where we delve into the science behind our research into cancer, therapies, and hope for a better solution. We're committed to sharing evidence-based insights and the voices of those advancing our understanding of the field of synthetic biology. To stay up to date with future episodes, subscribe to our YouTube channel, and if you have feedback, questions, or personal stories you'd like to share, we welcome you to reach out. You can find us on Instagram at michigan.msbt. Speaking with Dr. Biteen were Arnav Sharma and Jack Collier, members of MSBT, and I'm your host, Daniel Yang, and I thank you for joining us today.